Welcome to Let's Get to the Marks. We're looking at an A-level biology question on cell fractionation and microscopes. Please tick a box next to the sequence that shows the correct order of magnitude of these measurements. Pause the video, have a go, then unpause and watch the explanation. Okay, are you ready for the correct answer? The correct answer is row number 2, so put a tick there, and I'll go through now and explain how I got to that. So first of all, I've written these down into decimals, so not in their standard form. So 0.05 millimeters, that's 5 times 10 to the minus 2. Now, the next unit is in meters. If I want to also convert that to millimeters, you've got to times meters by 1,000. So that gets rid of, so that's basically 0.5 times 10 to the minus 2. So that's the equivalent to 0.005. Then I've got micrometers. Okay, I want to convert those into millimeters, so I need to times by 1,000. So I've got 0.5, sorry, divide by 1,000 because we're going back up to millimeters. So you divide when you go up, and that equals 0.0005. And then I've got nanometers, and I've got 50. So I need to divide by a million, basically, by 1,000 times 1,000. So I need to divide by a million. And that puts them all into millimeters. So what I've done is converted them all into millimeters at the bottom there. So you can see they're going from largest to smallest. And so there you go. That's how we clocked onto that one. We had to go through, convert them all to the same units, and then only then can you see clearly if they're in the right order. Okay, moving on to the next question. Pause the video, have a go. You've got to label identify what is D and what is E on this electron micrograph. So what is structure D? Well, it's pointing at a stack of thylakoids. So you can either say thylakoids or a stack of thylakoids or the thylakoid membrane, because it's hard to differentiate if it's pointing to the whole thing or a group, or you can say granum or grana, singular or plural for a stack of thylakoids. Okay. They might even accept thylakoid membrane because it's hard to tell whether it's pointing to the membrane or the whole thing, or just one individual disc. Of course, here you've got intergranal lamellae, but this round lump here for E, what is that round circle? You might be tempted to say, oh, is it a loop of DNA? Because you do find loops of DNA like you get in prokaryotes, but it's not. It's a starch granule or a lipid droplet. And so that's the second answer. Let's move on. All right, 5.3. The detail shown in figure 3 would not be seen using an optical microscope. Can you explain why? Pause the video, have a go, see if you can write down 2 reasons why a light microscope or optical microscope wouldn't be able to see all those different structures like thylakoids and intergranal lamellae. And basically, light microscopes or optical microscopes are limited by the wavelength of light, which is like 200 nanometers. So anything smaller than the wavelength of light, they can't distinguish as separate objects. So we say they're limited by the wavelength of light, which of course, if you were comparing to an electron microscope, which you don't need to here, but is a lot larger or a lot bigger wavelength than a beam of electrons that an electron microscope uses. But the main points here to get the 2 marks is just that limited by the wavelength of light, so they have low resolution. And if you want a definition of resolution, you don't need it here, but it's the ability to distinguish 2 objects as separate, okay, and not see them as one lump. Great. So the main points is optical microscopes are limited by the wavelength of light, so they have a low resolution. Basically, a lot of those organelles are smaller in their width than the wavelength of a light ray. Right, name an organelle found in a chloroplast and a prokaryotic cell. So again, you might be tempted to say circular DNA. Don't do that. That's not an organelle. It's a 70S ribosome. Okay, moving on to 5.5, we have one of these biology maths questions for 2 marks with a few steps to it. Once again, if you hit the like, that'll massively help me out. Make sure you pause the video. Unpause, check the walkthrough. Bosh! Let's go. Scientists determined the volume of a plant cell and the volume of the organelles contained. They found the volume of a plant cell is 17,500 micrometer cubed. The volume of the mitochondria in a plant cell is 262. The volume of all the mitochondria in a plant cell is 262.5 micrometers cubed. The volume of all the mitochondria and all the chloroplasts in a plant cell is 44.1% of the volume of the plant cell. Use this information to calculate the volume of all the chloroplasts in the plant cell. This should take you 2 minutes, 2 marks. Lovely. Okay, I'm going to go through the answer. So we know the volume of the plant cell is 17,500 micrometers cubed. However, they tell us here that 41 or 44.1% is the mitochondria and all the chloroplasts. So first of all, let's find out how much of the volume of the plant cell is mitochondria and chloroplasts. So we do 44.1 over 100. You can just do 0.441. And that is going to get us the answer. Smash that in the calculator. And that is going to get us 7,717.5 micrometers are all the chloroplasts and the mitochondria. Now we just want to know the chloroplasts, so we want to just take away the volume of the mitochondria. Oh my days, what am I doing here? It's like watching a 90-year-old flick through a newspaper. Okay, here we go. There you go. 7717.5, the total volume of the mitochondria and chloroplast minus the mitochondria gives us the chloroplasts on their own, which is what the question wanted us to do. Nice and easy. Hope you found that one easy. Normally the first mark's dead easy. So if you just come up with 44.1% times 17,500, you'd have got the first mark. Sometimes the second mark's really hard and needs 3 or 4 steps. This is quite an easy one. Check out my other maths videos like the one in the link here, where I go through a whole series of maths questions for A-level biology. All right, guys, hit the like, subscribe. Let's get to the marks. We're on to 5.6 here. A biologist separated cell components to investigate organelle activity. She prepared a suspension of organelles in solution that prevented damage to the organelles, described 3 properties of this solution and explain how each property prevented damage to the organelles. And it's worth 3 marks. So what 3 things did she do and how did it prevent the organelles getting damaged? Well, this is all part of your cell fractionation topic, so you should know all of this off by heart. It's part of the AS biology, but also obviously it's part of the A-level content. So number 1, use an isotonic solution. You can describe that by saying use a solution where the organelle and the solution around it are at the same water potential. And the explanation, this is going to prevent the net movement of water by osmosis, which will prevent organelles swelling up and bursting. Okay, for example, if you had a higher water potential outside the organelle and lower in the organelle, water will move in by osmosis and cause the organelle to burst. And we wouldn't want that when we're trying to study them. So that's your first mark. You only get 1 mark. I might put 2 ticks, but you only get 1 mark for each of these, so be aware of that. Property number 2, these can be in any order as well. They're not, you don't have to memorize a specific order. So property number 2, we use a buffered solution. Now, a buffer obviously minimizes changes in pH when you add hydroxide or hydrogen ions, but essentially it prevents enzymes or proteins losing their tertiary structure or their 3D shape. So enzymes don't become denatured, or other important proteins in the organelles do not become denatured, because most proteins only maintain their 3D shape in specific pHs. So property number 3, we want ice cold solution. So we want it really cold, and this is to reduce enzyme activity. And that will get you the 3rd mark. Obviously, you don't want enzymes going about their everyday business, especially if those enzymes will digest parts of the organelle. You might have proteases and things digesting proteins within the organelle, and we don't want that to happen, so we stop the enzymes from working, essentially. All right, guys, sharing's caring. Hit the like, punch up the bell, subscribe. Let's get to the marks.
You're still here? It's over.